results and for each of the presenters, they will have 3 minutes for presenting the thesis and 7 minutes for the Q&A sessions. There are 4 jurors that will be judging the presenter today. The first one, Ibu Siti Daniela Arwan, PhD, Indonesian Education and Culture Attaché in Bangkok. Dr. Muhammad Fitrah Alfian Rangga Sakti, MS, Researcher in Theoretical Physics at Chulalongkorn University. And Hana Nur Afifah Yogar, LLB.MA, Doctor of Philosophy Graduate Research in International Development Studies, Chulalongkorn University. For the technicalities, Kavini will present something. Kavini, over to you.
Okay, thank you. Okay, wait a moment, please. And I will share something. Okay, it's just like a recap. So this is the poster that we encountered during the technical meeting. So I would just like to highlight some points that are mandatory for you. First, of course, your title and the authors and everyone that included into your work, and then the logo of our embassy in Bangkok, and then PPI Thailand or Permitha, and then your affiliations here. And then don't forget to refer for your work to any paper or yes sites the information that you are going to use properly. And then if your work is funded by some foundation, then please acknowledge it properly. So this is just an overview. So this is a one-page poster, single format page poster. And then in this case that you are not allowed to scroll up or down or to zoom in or to zoom out because there is no necessity to do so. So by doing this, you can point out what kind of information that you would like to deliver, but just point it out. That's it. Thank you.
Okay, thank you, Kavini. Without further ado, maybe we will start for the first presenter. Abi Yazid Al-Bustami Rofi'i, are you ready?
Yes, I'm ready. Can you hear my voice?
Yeah, clear. Okay, the stage is yours. 3 minutes start from now.
Okay, good morning. My name is Abi Yazid Al-Bustami Rofi'i, PhD candidate from Faculty of Nursing, Chulalongkorn University. Have you ever seen other people who have difficulty getting up from a chair? This is a common struggling faced by those living with osteoarthritis, a chronic condition affecting millions worldwide. For many, osteoarthritis causes pain, stiffness, and reduced mobility, leading to significant physical limitation. In my research, effectiveness of nursing intervention enhancing physical function in people with osteoarthritis, a systematic review and meta-analysis, I explored how nursing intervention can improve physical function in individuals with osteoarthritis, particularly older people. Nurses play a crucial role in managing osteoarthritis, offering non-pharmacological treatment that aim to alleviate symptoms and enhance physical daily functioning. However, there has been a lack of comprehensive reviews in the effectiveness of the nursing-led intervention, especially regarding physical function. To address this gap, I conducted a systematic review and meta-analysis following PRISMA guidelines. The study analyzed 10 research articles focusing on interventions such as exercise, self-management, education, counseling, massage, and heat application therapy, all delivered by nurses or in multidisciplinary teams. The results were encouraging. Exercise interventions such as isometric and walking exercise showed significant improvement in physical function for people with osteoarthritis. Self-management program also empowered patients to better manage their symptoms, helping them maintain independence in their daily lives. Our meta-analysis revealed a statistically significant effect size, confirming the overall positive impact of the nursing intervention on physical function. While there were some variation among the studies, the effectiveness of nurse-led intervention was consistent. In conclusion, my research underscored the essential role nurses play in improving physical function for osteoarthritis patients. By incorporating this intervention into routine care, we can help reduce the burden of osteoarthritis and enhance the quality of life for millions older people struggling with this condition. Thank you very much.
Okay, thank you, Abiyazid, for the presentation. Next, we will move on to the Q&A session. For the juror, we will ask questions directly to the presenter for 7 minutes.
Pak Abi?
Yes.
Yes, very interesting research, and I wonder if you anticipate, for example, the condition in Indonesia, how this research will be implemented. Maybe I have 2 questions. So, how you define the older people in your research, and the second one, how older people in the village area or rural area can benefit from your research. I mean, with the limitation of the health worker in the rural area. I think that's my 2 questions. Thank you.
Yes. May I answer directly? Okay, thank you very much. So to define the older people, we follow the guideline from Ministry of Health that older people will be a person with the age over 60 years old. And for the in case of the older people in the rural area, my project will incorporate home-based exercise. So my project for now is try to develop home-based exercise. So to enhance physical function in people with older people with osteoarthritis. So we would like to enhance the physical function by engaging the older people to do the exercise in their own home setting. So that's maybe hopefully it can be answer the problem of the condition in rural area. That is my answer. Thank you very much.
If I may have one more responding to your answer, because you know that recently we have new government, and one of the priority programs is how to provide the health worker equally to all of Indonesian area. As we may know that the geographically is quite challenging. So is that what you mean, that home-based service is utilize the people, or is it to send the health worker, I mean certified health worker to go to the house, or how? Can you please a little bit elaborate on that? Thank you.
Thank you very much. So our project is home-based, highlighted home-based exercise. So it with the limitation of the healthcare worker, maybe in the rural area. So we would like to encourage older people to exercise by themselves in home setting. So maybe we already have a program like Prolanis, program pencegahan penyakit kronis. They can meet maybe one in a month, and then we encourage older people to do some activities in their own home. So with the limitation of healthcare worker, it can be done with the home-based exercise. Thank you very much.
Thank you, Pak Abi.
May I jump in, Naila? Okay. Hi, Mas Abi. Thank you for the presentations. I just would like to have some clarifications or probably questions, either questions or clarifications. You mentioned that you would like to highlight home-based exercise, but actually I couldn't find that statement during the presentation. So I'm just wondering if that's kind of the finding or ongoing research that you're going to apply at the end of your research. And as you also answered the questions from Ibu Daniela, I think I would like to suggest you to have some kind of statement to limit your research, some kind of limitation. So you're gonna have some kind of space for people to do more research that that correspond with your current research as well. And another comment about technical. So it's more not questions actually, more like suggestions. And another comment, more like technical comments. I couldn't read your figure. It's kind of small, so I don't know what is actually in there, even though I have checked earlier. But I think it's really important to show to the audiences because you talk to the people, right? So you just want to know that the presentation is grasped by the audiences as well. And the last one is probably because you are the presenter, so you know better about the presentations, but probably some people also have to know the understanding or the definitions of the some kind of jargon technique or technical terms. Your presentation contained so-called osteoarthritis. So probably some people may heard about that, but maybe you can just give some kind of highlight definitions about that. But overall, yeah, I enjoy your presentations and good job. Thank you.
Yes, thank you very much. So this presentation is my preliminary studies to develop what I call what I mentioned previously, the home-based exercise. So what I present today is the preliminary study, the systematic review and meta-analysis to develop further intervention to older people with osteoarthritis. Yes. Thank you very much.
Okay, the time is up for Q&A session. Next, we will move on to the next presenter, Islah Mutakin. Are you ready? Okay.
Yes. Can you hear my voice clearly?
Okay, clear. Yes. The stage is yours. 3 minutes starts from now.
Okay, good morning, everyone. I'm Islah Mutakin from Chemistry, Chulalongkorn University. So imagine something so tiny you cannot see it, yet it can cause lifelong damage, like brain and kidney damages, motion disorder, and also even death is deadly. This something called as mercury. You know, mercury known as a highly toxic metal that can exist in our environment at incredibly low concentration. And you know that it's oftenly undetected by the naked eye. So mercury contamination is a global issue, and if not properly managed, it poses serious risk to human health. That's why organizations like the World Health Organization has very strict limits on how much mercury is allowed in drinking water. To give you an idea, this limit is as low as 1 ppb, and like trying to find a single drop of water in an Olympic-sized swimming pool. The challenge is, how do we detect such tiny amount of mercury in water or even inside our bodies? Conventionally, scientists have used high technology instruments to do this, but these methods are expensive, complicated, and require trained experts to operate. They also cannot be used to easily detect mercury in living cells, which is important for studying how mercury affects human health. That's where my research come in. I've developed a new way to detect mercury that is simpler, cheaper, and more effective. I design and create a special person chemosensor that lights up when it's come into contact with the mercury. We can think about a glow stick. It stays off until it detects mercury, and then it glows brightly, making it easy to spot the contamination. Okay, next, let me explain how this probe will interact with the mercury. Imagine this probe as a thiourea. It's contain the solver, and it needs we can conclude it as a lock that needs a special key to open. In this case, mercury is act as the key, and when mercury unlock the thiourea by removing the sulfur here, it will produce or enhance the fluorescence enhancement, will produce the urea. And what makes the sensor even more exciting is the ability of the sensor. So I've tested in real water sample, including the pond water, drinking water, and also the tap water, including the application in cell imaging. So it works incredibly well. It means it could be used in future biomedical application to track mercury in the body without harming the cells. In short, my research offers a new tool that can help protect both people and environment from dangers of the mercury. With this probe, we can identify mercury contamination early, keep our environment cleaner, and hopefully prevent serious health problems in the future. Thank you.
So the viewer can ask directly to the presenter.
Mas Isla.
Yes, Ibu.
Yes, thank you for your presentation and the future impact of this. And maybe my simple questions is how much or how big or how much do you think we can reduce the cost by implementing your research? And I assume that this research will also be we can also implement in country in Indonesia. So I'm not sure how far you go on this research, but what will be your anticipation in order we can adopt or we can implement your research back in our country? So I think that's my two questions.
Okay, thank you for the question, Bu Daniela. So actually, for if you're asking about the big deal about like this research, so to overcome the limitation of the previous method that I mentioned, like it's using the instrument that we need to provide it, the instrument that maybe it's really expensive, around like maybe in rupiah, maybe 1 billion or maybe more. So by using this alternative, as a chemist, organic chemist, we synthesize the material that can apply it directly to the contaminant. But we also use the instrument, but that instrument is not that as expensive as the conventional method. Maybe it can reduce 10 times for the cost. And for my research is actually it's a preliminary result that we propose the material that can give the highly selectivity and sensitivity for the mercury itself for the detection. And we designed because in my field is to find which material that really give the highly sensitivity itself. And our purpose is to detect it in the water sample. So if we can adapt it to apply it in Indonesia. Maybe it can be we can propose it like develop the device using this material, and then we can detect it directly in the field of the environment, like maybe in the wastewater or something like that. I think that's all from me.
One more. Where is your I mean your field of what should I say? Do you do the research back in Indonesia? I mean for the sample or everything, or you do here?
No, I've done
the testing.
I've done here actually for the real water sample that I've tested is around my university. So I just spiked I've just took some sample from around the university book. So just for the sample.
Thank you.
Yeah, thank you.
Okay, I would like to ask a question. Is my voice clear?
Yeah.
Hello? Oh, okay.
Yes.
Okay, thank you. So, can you tell me again what's the material that you use for the fluorescence?
So actually, it's a small organic compound. Because in my field, it's focusing on the organic compound. But for some different, for another material, they use another compound like quantum dots or even like some metals. But in my cases, I use the small organic compound like the new one, the novel compound that I've developed.
For the future study, maybe if we want to detect like the mercury on our blood, can it be used? Because I don't know the material, maybe it will react with the blood component or something, maybe.
Okay, so actually for the interference, we've studied in like my probe is competed to for example, we prepare the sample containing not only the mercury but also the interference like another metals. But as you mentioned about the blood, we haven't tried it yet. But for example, in the blood we have the iron, right? Iron. But in iron we also can be use it, but we don't know about the another containing contents in the blood. It will interfere or not. So we haven't tried that far.
Okay. Okay, thanks.
Yeah, thank you.
Naila, I have no comments. I have written comments, but probably I'll just deliver it later on. Thanks.
Okay, the time is up for Q&A session. Thank you so much, Isla, for the interesting presentation. Next, we will move on to the next presenter, Kati Febriliani Rahayu. Are you ready?
Am I audible? Sorry.
Yeah, audible. Okay. The stage is
wait a
yours,
minute.
starting from now.
Okay. Ladies and gentlemen, imagine this. Every time you take a sip of water, you could be ingesting of thousands tiny and invincible plastic particles called nanoplastic, originating from the breakdown of everyday plastic items like packaging materials through UV irradiations, chemical oxidations, and mechanical fracturing. Nanoplastic now can be found in our air, water, and even our blood. And due to their tiny size of less than 1 micron, nanoplastic can carry toxic chemicals, penetrate to our blood-brain barrier, and trigger inflammations in our brain. So this makes the removal of nanoplastic is not just important, but it's very essential. Removing nanoplastic from water is no small feat. Traditional methods like adsorptions using biomass-derived adsorbents often require a high temperature and pressure for fabrications. Don't worry, there is a hope. So who here likes seafoods? And you're probably familiar with the green mussel cells, right? So in 2021 alone, Thailand produced up to 52,000 tons of green mussel cells, with 70% of it ended up as waste. But the good news is these green mussel cells are composed of 78% of calcium carbonate adsorbents, which can be extracted by simply adding a base solution and sunlight. And to make it even more effective at capturing nanoplastics, we modified the calcium carbonate with a cost-effective fatty acid like stearic acid for surface modifications. And how does it work? So nanoplastic, which have a long chain of polymer structure, will interact with the carbon chains of stearic acid through hydrophobic interactions, helping them to eliminate nanoplastics from water. And recently, we successfully prepared calcium carbonate modified with stearic acid at ambient temperature without the need for heat and pressure. And you can observe the successful preparations by testing the surface tension when at water drop it's added, and pure calcium carbonate has a contact angle of 0 degrees, while the modified version has a contact angle of 70 degrees. And the results, we achieved up to 98% removal of polystyrene nanoplastic 100 ppm concentrated one with just 2 hours stirring at room temperature. And we believe that this green approach aligns perfectly with the 12 principles of green chemistry, focusing on waste prevention and mild conditions, as well as the sustainable development goals for clean water and sanitation and life below water. Thank you.
Okay, thank you. Next for the Q&A session starts 7 minutes from now.
May I start? Okay. Hi, Kitty. Thank you for the presentation. It's very
Hi.
interesting. And I
Thank you.
gain new knowledge today. I just would like to know further a little bit from your perspectives or from your work currently. How do you engage with the people around you? I mean, like lay people or just like non-scientific people to work with you together to collect the GMS? And then how do you see this as a part of some kind of collaborations work with them as well? Because from what I heard from your presentation, it's more like the perspective from you as a scientist, right? But how do you collect the GMS itself, and then how do you make people understand, and what kind of benefit they will receive once they're working with you to collect some kind of so-called waste?
Okay, thank you so much for the questions. Actually, for our feedstock, we obtain it from Ampawa market, if I'm not mistaken, because it is like basically a project from our lab, and our lab is like so focused to convert calcium carbonate base derived from these green mussel cells waste to utilize to convert it into calcium carbonate and then utilize it for further applications. And I think actually I also did a presentation for a project. It's like a presentation for a project, you know, like a business competition. I did research, but it's like a literature research in Indonesia, right? Because in here I work in Thailand, but I did a little literature research in Indonesia. So many as well, like green mussel cells waste has been produced per day, especially if the location, for example, like in Gresik, you know, like East Java, they are located near the how to say, the beaches, you know, and then they produce so many green mussel cells waste, up to like 7 kilograms in like per one store in there, like one area or one beach. So I think if I would like to empower those people, I need to engage with them in the more simple communication, you know, simple communications with them, and then make them to acknowledge that these real threat of nanoplastic is very, very serious problem. And actually, we can make it work with them. We can tackle those issues by doing it together, by also using their ways. And maybe in the future, if I want to collaborate with them, I would like to give them some incentives as well. So it can also enhance their productivity in terms to collecting this green Marshall cells waste, I think.
Cool, thanks.
Okay, thank you so much for the questions as well.
Okay, Mbak Katie, thank you for your great presentation. I wonder if you maybe elaborate further, as you mentioned about the SDGs. As we know, the SDGs will finish. I mean, will complete in 5 years' time, because this is already the end of 2024. So, how big do you think your research will impact on the one of the goals in the SDGs? Looking at the SDGs will complete in 5 years' time. That's my first question. And the second one, because I'm not from the field of your knowledge, in which area that can give more benefit if we implement your research. I mean, in a real life. Maybe that's my 2 questions. Thank you.
So the first question is like how big my research will impact, right? Okay. So I think, in my opinion, this research is very, very, how to say, it accommodates multidiscipline aspects, because first, I think to make project to make it a big impact, we have to see more perspectives rather than the research perspectives itself. It should accommodate like the economy and social and also environmental aspects. And this research, I think, it accommodates it all. First, we can also enhance the value of waste itself. We know that waste itself is not, it's like a trash. We cannot use it. But in this research, I would like to emphasize we can convert trash into treasure, something like that. And then for the economy, I believe that by utilizing waste that has been produced from the farmers, from the green Marshall cell spawn in that area, it can also encourage them to take actions in terms of engaging in more environmental activities, because my team and I will convert these waste into a material that is going to be beneficial for the environment itself. Something like that for the first questions. And how big it is, I think, since green Marshall cells can be found everywhere. So it's not only in Indonesia, right? So we can also find it in Thailand and maybe other countries as well. So I hope this can be utilized globally, not just a local area like in Thailand or maybe in Indonesia, but I hope it can be utilized globally. And I'm sorry for the second question. I didn't catch it. Could you please repeat it?
In terms of achieving SDGs goals, because
Yes.
SDGs will complete in 5 years' time from now. So yeah, because since you mentioned about SDGs, how convinced you are to contribute to the achievement of that goals in 5 years' time?
Okay. Actually, I'm taking a business. It's like entrepreneurial for chemistry because I'm from the green chemistry program. So I'm taking like entrepreneurial chemistry, and the Ajahn, he taught us about the technology readiness level. So I'm currently assessing that technology readiness level, since our lab has been developing this method or material for such a long time. So I think I'm quite sure that we can develop it for like within 2 or 3 years in terms to push it to the market, since we already have the foundation of the fabrication. So we would like to scale this up and also apply it for real applications.
Thank you.
Thank you so much.
All right, thank you so much, Kathy, for the presentation. Next, we will move on to the next presenter, Niar Ibrahim Rose. Are you ready?
Okay, so
Okay, clear.
sorry, can you hear me?
Yeah, the stage is
Okay.
yours, starting when you speak.
Okay, good morning, ladies and gentlemen. Hopefully this morning you already drink coffee because this moment I will present about my research about the spent coffee ground, which I change it to become the valuable product. So as we know, nowadays coffee already consumed by a lot of people, which for the scientists, we already know about the component of the coffee. It consists with the hemicellulose, which the main is galactomannan, which it can be converted to become mannose, which that is the monosugar of the galactomannan. Then why is it important for the spent coffee ground? Because it can be utilized for becoming some products. It can become prebiotic, anti-pathogenic compound, anti-cancer, monooligosaccharide, and then it can be converted to alcohol, sugar, and it can be used for the substrate for the enzyme. Then how it can be prepared for to convert the galactomannan to the bioproduct. So as I mentioned, it consists with the galactomannan. So there is enzyme that can be cut the structure of the galactomannan that called beta mannanase enzyme and alpha galactosidase enzyme. But in this research, it just focused for the mannanase enzyme, which the objective of my research is for screening the lab Aureobasidium species and then optimize the spent coffee ground to produce the monooligosaccharide and test of the monooligosaccharide for prebiotic. So how it can be answered of the objective. So for the first step, I did the screening of the Aureobasidium strains from my lab for producing the mannanase, which the best mannanase activity it will be used for the production. Then for this optimization of the spent coffee ground, monooligosaccharide, it was using the design expert for getting the optimum condition of the spent coffee ground monooligosaccharide. Then for the last, I did the prebiotic test with some of probiotics. And after that, I will get I got the number of the probiotic. So how my research, the result of my research. So from 40 strains of the Aureobasidium yeast, so I got the best one with the high activity of the mannanase with 8 unit per milliliter, which it can degrade the galactomannan at 35, 50, and 50 and 70 degrees Celsius. After that, this enzyme used for the producing of the monooligosaccharide. Okay, thank you.
Okay, thank you. Next for the Q&A session.
Okay, I would like to ask. So in this research, you do research on the coffee. What kind of coffee seed that you use? I mean, because we know that we have lots of types of coffee seeds, especially in Indonesia, for example.
Mm-hmm. So thank you for the question. So for the coffee that I use, I just got from the one coffee shop. So consistently, I take the coffee from the coffee spent coffee ground from the Starbucks that's near from Chula. So I just take from their coffee shop, and when I ask them about what is the types of the coffee, they said it's Robusta. So just only one type of the coffee.
Okay, so it may be different for different coffee, or
Yeah.
maybe. Oh, okay. Okay, maybe that's it for from me. Thank you.
But Niat,
Yes.
I think my simple questions is because I didn't read all of your research yet. So, what will be your thought about the final product of your research? Thank you.
So okay, thank you for the question. So for the final product, it can, as like my presentation, it can the spent topic ground can be a valuable product, which are the product that I already produced from one spent topic ground. I can obtain the manganese, the substrate for producing enzyme, and then can produce the prebiotics, and then it can be used also for the compost.
Thank you.
Hi, Niar. Thank you for the presentation. It's very interactive PPT. I just would like to suggest a little bit, since the questions were coming from Ibu Daniela and Mas Otpian as well. Probably if you consider it's important or probably worth thinking about how you can set the variables as well. Like, for example, correspond to the first questions or comments from the judge as well, asking about what type of coffee. And I think it's also important to see how the coffee itself is growing or coming from. Like, for example, maybe coffee from Indonesia and Thailand are pretty different. The types of coffee itself is different as well. It's probably just minor considerations, but I think it's really like showing how you really considerate on how determining the variables of your research. But yeah, good luck, and it's very interactive presentations. Thank you.
Thank you so much for the suggestion.
Naila, I give it back to you.
Okay, thank you. I think the Q&A session is already finished. Thank you so much, Niar Ibrahim, for the presentation. Next, we will move on to the next presenter, Ian Laurenza. Are you ready?
I'm ready. Could you please stop sharing the slide, please?
Niar, maybe you could stop sharing the...
Yeah, thank you very much. Could you see my slide?
Yeah, it's clear.
Okay.
The stage is yours, starting when you speak.
Okay, thank you, MC, for the introduction. My name is Yayan Laurenza from Department of Packaging and Materials Technology, Kansas University. Today, I would like to present parts of my thesis with title of Multi-Layer Food Packaging from Thermoplastic Starch, Citral, and PBAT, focusing on adhesive failure and citral release behavior. So biodegradable packaging now become a trend in the research and industrial field, considering the regulation on each country. For example, in Thailand,
Excuse me.
now...
My screen hasn't been changed. Is something wrong with my screen? Sorry. But I can hear the voice, but my screen hasn't been changed. Yeah.
Okay, ma'am. What about others? Can you...
Ibu Daniela, probably you have to click on the... Yeah, okay, cool.
Okay,
Never
yeah,
mind.
yeah. That's done. That's okay now. Okay.
Okay, you can see my screen right now, ma'am. Okay, so can I start from the beginning again, MC?
Okay, yeah.
So sorry. Okay, so again, thank you, MC, for the introduction. My name is Yayan Laurenza from Department of Packaging and Materials Technology, Kansas University. Today, I would like to present parts of my thesis with title of Multi-Layer Food Packaging from Thermoplastic Starch, Citral, and PBAT, focusing on adhesive failure and citral release behavior. So biodegradable packaging now become a trend in the research and industrial field, considering the regulation on each country. For example, in Thailand, now using single use of shopping plastic bag is not allowed. However, it has limitation on the properties of single biodegradable material, especially thermoplastic starch or TPS that are poor water resistance. Those multi-layer film could be a solution. So the objective of this research was to investigate the effect of modified starch and citral addition on the adhesive failure and citral release behavior from TPS PBAT multi-layer film. In this research, we are using biodegradable material, which is PBAT as outer layer, and TPS, NS, and OS as inner layer, and citral as self-adhesive and antimicrobial compound. And here what we have found, in the point 3.1 here, we could see that TPS-OS monolayer film had fibrous and higher roughness than TPS-NS. This gives a significant effect on the adhesive build strength on the point 3.2 here, especially after citral addition, due to more mechanical interlocking was formed in the rough surface during lamination. For your information, adhesive build strength is the force to separate two layers in the multi-layer film system. It is also explained the failure behavior here in point 3.3. We could see that the more damaged surface was formed in TPS-OS with citral incorporation due to the bonding between layers was stronger than the stability of TPS-OS film itself. It is called cohesive failure, and it is causing TPS more attached or present on the PBAT build surface here after adhesive build test. Here detected in FTIR spectra on point 3.4 by intensifying the peak of OH group that belongs to TPS. And the TPS-OS had higher hydrophobicity than TPS-NS, and it's giving a different citral release behavior here that was observed by GCFID within 13 days. The antimicrobial citral had higher tendency to release from TPS-OS due to higher affinity and diffusivity, which could be more effective as antimicrobial active packaging. And this is the final output, the multi-layer packaging with citral active release for food packaging. So we conclude that the biodegradable multi-layer film was successfully developed, with the TPS-OS had more stronger bonding due to roughness and affinity that allow citral to release from the multi-layer film. So this is the end of the presentation. Thank you for your kind attention.
Okay, thank you. Next for the Q&A session.
Hello again. Thank you for the presentations. My question is very simple. I think once you've done the research or you are now finishing your research, or I don't know what stage of your research actually, but I think at the end of the day, once you have collected the like all the data and then processed that you have the product that you are aiming to have, I just wonder what is your expectations or how do you see your product to be, I don't know, like marketed, or how do you communicate to people that, oh, my product is, let's say, have better quality and then it's coming from blah, 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 and it's better, let's say you're simply saying better quality than other products. And then how do you engage with them? How do you communicate with them? And then how do you kind of like market your product? And then I think if you engage with the market system as a whole to promote your product, how do you engage with other stakeholders? For example, how do you convince the policymakers to use your product, or maybe just simply asking the producers or compete with the current food packaging producer to have some kind of better place in the market system? Thank you.
Okay, thank you for the very nice question. So actually, if we bring the biodegradable things to the market, it will give a good impression to the consumer. So it will be good also to the packaging or the food company maybe that will be the user of the product, of the end of this product. So we can communicate it with the HRD in the company. So we will convince them that the price, the cost of the materials, the cost of production, and the processability in the industrial scale, is it very possible? And because we are in this research, I am using the industrial scale, not industrial scale, I'm sorry, industrial equipment in the pilot scale, but it is easily adapted in the big scale of industry. also will be a good information to the company that will be a user of our product. And for your information, actually our department is open for consultation, yes, or discussion from any other institution. Actually, there's some company from Indonesia that come to our department last time, and they are from the company that who want to develop the biodegradable packaging. They are asking how to develop a material, which material that are good for this product and that product. That also will be very good connection for me to. It's also good connection for me too. When I go back to Indonesia as well. And for I think that's answered the question, right?
Yeah, it's cool. Thank you.
Yeah, you're very welcome.
Mbak Yayan?
Yes, ma'am.
Yes, I understand that from your explanation about the cost incurred in your product and how big do you think the possibility of having this product in Indonesia? As we know that most of us, we may be in the market or in a traditional market, still have a big chunk in our country. And as we may see that the packaging is also very simple, because maybe our main our main goal is still to have cheaper products. So how big do you think it can be implemented in our country for your product?
Yeah, okay.
Thank you.
Okay, ma'am, thank you. So in this project, we are using starch that easily you can find in your kitchen or in the market. And all of you, I'm sure you know the starch, right? That normally for baking or making a food that really cheap actually. And it's prepared from that. It's made from starch actually. And the properties is already good. And for the pipette itself, or polybutylene adibate tetraphthalate, but actually it's already commercial. And once I went to Bali and I went to Krishna, if you guys ever to Bali and you guys know the souvenir shop, the Krishna in Bali, yeah, they are using pipette. I recognize it easily when I got that bag, and which mean that the biodegradable pipette materials is already implemented in our country. It's already started. Yeah. However, it's still like have some need more improvement. Very depend on the application. In that case, when I went to Bali, the plastic bag is for shopping bag, biodegradable shopping bag, and it is reused. Even because pipette is biodegradable, it have long shelf life, long durability, so it's like not a single use. It's like you can use many times actually. And for food that are contact with food, normally we need improve the properties. For example, the multi-layer like this. And so the cost is really possible. It's already started actually. Even in Thailand, if you ever went to Amazon cafe, I'm sure you know Amazon cafe. And when you take it away, your drink, and you will get the plastic bag, the small for the cups. That's also from pipette. I know that. It's really typical of pipette blend with others called PLA, the material. It's the so it's since it's already implemented, already scaled up in industrial scale. Of course, the industry already make a plan about the cost, about the industrial scale plan and production efficiency, and so many. They plan it already, and it's going to market already. So the price will be fine. Yes, ma'am, something like that.
Thank you.
My pleasure, ma'am.
Okay, maybe I'm the last. I will ask the last question. It's actually only to clarify. So in this research, you use three different materials, from the starch, citral, and pipette.
Yes, correct.
Because in some, I don't know, maybe I'm mistaken. So in your presentation, it's like you only use starch and citral to put on pipette, or?
Oh yeah. I'm sorry. Actually, I need to explain this first, right? So I have two layers here. The first layer is TPS TPS film layer, and the second layer is outer layer, is pipette layer. So both of them are different layer, not blending. So we have two layers that we laminate together, and the citral is in the between of layers. So the structure can be like this. If you can imagine, the dark blue is the pipette layer, and the light blue is the TPS layer. It is like animation that you can imagine, and the citral is the yellow dot here. Yeah, inside the layers. Actually, citral have two functions here as adhesive, because the bonding strength is increased here after citral addition, and also it can be released here by GCFID. It can release, and it can be preservation agent, antimicrobial agent. So yes, correct. I'm using three materials different, and they are not blend. They are just different layers. Because in the past, I used two or three materials in my previous project. I'm blending it, and still the TPS presence on the surface of the film, and it still react with the moisture in environment, and the barrier properties is still limited. It's still not good. So by preparing with multi-layer like this, I got the better barrier properties, thing like that.
Okay, thanks.
Okay,
My pleasure.
the time is up for the Q&A session. Thank you so much, Yayan Lorenza, for the presentation. Next, we move on to the next participant, David Nugroho. Are you ready?
Yeah. Yep. So everyone can see.
Okay. The stage is yours. Starts from when you speak.
Okay. Good morning, everyone. So today I would like to present about what I do for the cleaning the antibiotics problem in the environmental. As we know that the antibiotic pollutant is like one of the kind of the serious environmental pollutant in the world. So how to fix this problem? I'm just thinking that, okay, I mix some material that is also environmental safe for degrade or photo degradation of pollutant in the water waste treatment. So in this project, I collaborate with a Korean professor that I make the M-accents. I mix with the zinc oxide and cadmium selenium that is with the low of the concentration. And then it showed the result that my catalyst powder or my powder can be degradation of the various of pollutant, example ciprofloxacin, amoxicillin, and ofloxacin antibiotic can be until 99%, but for the ofloxacin is only for 98.6%. And then maybe like some of you that study about the material also in the research, we must make sure the characterization of the catalyst powder, is show the characterization under SEM, TEM, including XRD, that for the mixture of the catalyst powder, that is make sure that what I want to make, because the novelty of this research is I'll make the new catalyst powder for photo degradation application. And it was show result that my catalyst powder can be decreased 99.9% for ciprofloxacin, 99.9% for amoxicillin, and 98.6% for the ofloxacin antibiotic. So it's mean that this catalyst powder can be used for multiple of antibiotic pollutant, can be decreased antibiotic pollutant in the water waste treatment. So in the future, now I collaborate with the hospital in the Konkan, it means the Srinagarin Hospital, for develop in the real pollutant, hospital pollutant that real contain of the antibiotics. And in the future also, I also do the development for, we want to know that my catalyst powder is for test for the carcinogenic and another things about the human safety. So is want to make sure that my catalyst powder is really safe for environmental, and also have the good things application for photo degradation of pollutant also. Thank you.
Okay, thank you. Next, the tutor might ask the questions.
Hi, David.
Yes,
Thanks for
hi.
the presentation. I think as you mentioned that you're currently working with one of the hospital in Konkan province, right? I just would like to know that if your research is I'd say it's applicable or doable in every hospitals, let's say, would it be possible to have some kind of people like scientists like you also working with the other hospital as well? I'm just trying to foresee the possibility or doability for doing this kind of work, because I think like you're specializing in this area, so I'm not sure if there will be people as well. It's more like human resource that who gonna work on this one. And then as a very technical questions, how about the cost that you're working on this one? Like who gonna fund this? Is it part of the hospital's project as well, or part of your academic project that is implemented with a hospital?
Okay, so my project will be easily for applied, especially in the hospital, because now what I want to try in the hospital, because now I'm just using the DI water. I mean, like just only water without any contamination, just pure the pollutant that I want to know. So in the future, I want to really know that is it really work for the applied in the hospital waste, something like that. So after, is it possible can be decreased with the 99.9% if you, I mean, like have the optimum condition one. So is it gonna be easy for apply for degradation? Because like photocatalytic degradation, I also using just only the visible light, just only like need for the steering one for the degradation of the antibiotic until 240 minutes. And then how much the cost of this one? The cost is this cost is not too much expensive. I mean, like, yeah, maybe like some of my friend in here, such as like material researchers, know the price of like MX and everything, because like this research cost is based on my thesis, not any funding from the hospital.
Cool, thank you.
Mas David, thank you for your presentations. I just would like to know your literature review about your research. So from your literature review, how big in scale about the antibiotic pollutants that you find in your literature review, and how much it can be reduced after you finish your research and we implement your research result on that pollution. Thank you.
Okay, ma'am. So thank you for the question, ma'am. The literature review told me that the mostly the concentration of the pollutant in the wastewater treatment is around 15 ppm until 20 ppm of its antibiotic. So in this research, I do like 20 ppm part per million in the concentrate of the pollutant. So in this research, I using various of, actually have the detailed data in my publication, but the maximum of my catalyst in this research I do for the 20 ppm, 20 part per million, and it still can be decreased 99.9%. And then another thing, the stability of this powder also can be used for like 5-time recycle. In this research I using, so after I using the first degradation, first pollutant, I dry, and then I use the same powder. I didn't use the new powder, I used the old powder, and it still can be used for the recycling until 5-time recycle. But in the future, maybe can be improved for like 10 times or 20 times. We can see like the efficiency of the powder.
Thank you.
Okay, thanks for the presentation. I would like to ask about the sonoelectron and photoelectron. Can you elaborate more about that?
Oh yeah, actually the photocatalytic is just only used for the light, visible light or like UV light. But for the sonophotocatholic, I using the vibration also. So I using the ultrasound to help for degradation of the pollutant. So we call sonophotocatalytic. If photocatalytic just only like we steering under the visible light or UV light, but sonophotocatalytic, I use the ultrasound like ultrasono for helping for is more faster for degradation compared than photocatalytic. But now, during my another research, I developed the new. I using to say that magnetosonophotocatalytic. It will be more promised result because I using magnetic effect and then sonophoto and then the sono and then the photocatalytic.
What's
Because
the
now like we want, yeah. Because now, pardon, sorry, sir.
what's the source of the vibration for the sono?
Oh, in this research, I just using the ultrasound nicoter. So I put the Becker glass in the ultrasonicator, like sonicator box, and then I put UV light, visible light, and then steering, and then just like checking every 10 minutes, and then got this result. And then for the check the concentration, I check by UV spectrophotometer for know the concentration of the pollutant.
Okay, thanks.
Sure.
Okay, that's wrap up our Q&A session. Thank you so much, David Nugroho, for the presentation. Next, we will move on to the next participant, Dini Aulia. Are you ready?
Oh, wait a moment.
Okay. Okay, clear. The stage is yours, starting when you speak.
Hi everyone, my name is Tini Aulia. Before I briefly start to explain about my project, I would like to introduce you guys about the dengue treatment limitations. So, dengue fever is the most important mosquito-borne disease in the worldwide, especially in the tropical countries. And now we have available dengue vaccine in the world, but they come with the limitations such as age restriction and also expensive. Besides the vaccine, we also need anti-dengue drug, but unfortunately, until now, we don't have anti-dengue drug available in the market due to the toxicity issue and unwanted side effect. So moving to my research overview, so my research comes from the previous anti-dengue drug development. So the drug can inhibit the viral enzyme very well, but the drug also inhibit the human cells. That's why it leading to the toxicity. And here our research goal is to discover new untoxic drug that can selectively inhibit the viral enzyme but didn't disturb the human enzyme. So to reach this goal, we have 4 strategies. So the first one, we design the novel compound by modify the structure of the natural substrate of the enzyme. And then at the end, we have 200 design of the compound. And then we perform the molecular docking to know the interaction with the enzyme. And we select the 20 structure of the compound to synthesize in our lab. And after we synthesize the compound, we confirm the properties by some instrument. And here we go to the biological testing. So we testing the synthesized compound against the viral enzyme, the virus, and also the human cell to know the toxicity. And I can say from among the series, the compound, the XOA23 is the best compound because as we can see, the compound show viral, the inhibition of the viral enzyme almost 100%. Also, the inhibition against the virus almost the 80%. And interestingly, the compound show the cell viability against normal cell almost 100%. It means the compound it's really safe. And also non-toxic for the human. So in the conclusion, so I already obtained the potent compounds against dengue virus, which is the compound the XOA23, and we will do the further research to know and to obtain the real anti-dengue drug in the future. Thank you so much.
Okay, thank you. Next, the juror might ask questions.
Dini, Thank you for the presentations and for the interesting research that you do. Have you found the efficiency of your innovative drugs after you do the research, and also the availability in the future in the market, so we can easily get and easily access? I think that's my question. Thank you.
Thank you so much, ma'am, for the question. So for the efficiency of my potent drug, so first we test against the human cell first. Like we say this in vitro testing. After we pass the in vitro testing, so testing against the cell, we continue to test against the mammal, such as the rat. We call this the in vivo, but in my project now, we just test for the in vivo first. After we got the potent compound against the cell, then we can continue to the in vivo cell, which is for the rat. And for the availability in the future, so actually the drug discovery, we have very long step to bring the drug until published in the market and cost a lot of money. So yeah, my project here is really like early step for the drug discovery. So after, and we need a lot of collaboration to reach our goal until we can market the drug in the future.
Probably because this is your early stage, maybe your expected result, your expected outcomes from this research, because you start with, because it's expensive, hard to find, it's not available in the market. Then what will be your expected result? I mean the advantage of your research that you expect to have. Yeah.
So I expected we have a good antiviral activity for my compounds, and then we can publish our work, and probably in the future we also can test in vivo. So yeah, if it expected like how can I say, if it succeed, and then yeah, I expect to have FDA approved drug in the market, but it's quite a long time to do. But so yeah, and now for now our goal is to find the compound that can show good activity against the cell first.
Thank you.
You're welcome.
Hello, Dini. Very beautiful slide and engaging presentation as well. I have a question. Well, let me just reframe my questions, because you just mentioned that it's kind of like preliminary stage of your research. But how long did you wait for the research ethics committee to give you a permission to do this kind of experimentation?
So actually, I have another collaboration from another department, also another university. So for testing the compound to the cells, to do the in vitro testing, we have collaborators. So I didn't do it by myself. So yeah, so I have collaborators to do it for me.
Oh, okay. Cool. So second question, probably just moving a little bit further on that issue. I know you've been doing experimentations with other collaborators as well. I'm just wondering, when you refer your study or your experimentation, do you see any similarities in cases in other countries as well, that you can, I don't know, like you are referring to that cases so you can come up with this kind of innovations, especially when we encounter with the issue of dengue fever, for example. I don't know about other tropical countries because probably Indonesia is one of the most susceptible dengue fever countries. So I'm just wondering if you also have some kind of references that you referred so far during your experimentation. That's it, Dini.
Sorry, I couldn't catch up your question. Can you repeat again?
No problem. So during your experimentation, do you have any references, like seeing other cases in other countries that are also doing the same thing with what you are doing currently? Just wondering because not all countries are tropical countries, but I think it's worth considering to see other cases as well.
Okay, I get it. So yeah, many researchers doing same thing like me to develop the anti-dengue drug, but actually a lot of protein to target the dengue virus. So here I pick the NS5 protein. So other researchers maybe they target another protein in the dengue virus, but another researcher in another country, they modify this compound, the SAM compound, like they directly modify the structure. But for me, I only pick the center part of the molecule, and then I modify the rest, the left part and the right part. I do this because I know if I modify the whole structure, and then it will leading to the toxicity for the human cell. So I just pick the center part to give some same interaction with the enzyme, and then I modify the rest. So this is my novelty, the novelty for my research, that another researcher didn't doing same like me.
Cool. Good luck.
Okay, thank you so much for the Q&A session, and thank you, Dini, for the presentation. Next, we move on to the next presenter, Steven Dwi Purbantoro. Are you ready?
Yeah, can you hear my voice and see my screen?
Okay.
Okay. So good morning, everyone. My name is Steven Dwi Purbantoro from Faculty of Veterinary Science, Chulalongkorn University, and today I'm bringing up my study about osteogenic growth peptide, a potent peptide for bone regeneration. So first of all, I would like to bring it up about the inflammation of the gums, and yet that you may experience before this. If it's left behind, it will cause more severe condition into the deeper tissue. It's called periodontitis. And in addition, if it's getting left behind again, it will cause bone loss, and it will lead to the higher risk of the pathologic fracture, or it's the incontinuity of your bone. So right now, it's not only happening in humans, but also in animals. And right now, the current trend of using tissue engineering approach is proposed to treat this periodontitis. Tissue engineering itself the aim is to regenerate the surrounding tissue to substitute the defect. And right now, we specifically to regenerate the bone tissue for periodontitis case. Essentially, it has three components: the cell, scaffold, and also signaling molecules. And right now, the signaling molecules have been proposed to induce more because it directly triggers the activation of the cells. And OGP, or osteogenic growth peptide, has been proposed because it has great potential to treating periodontitis. It's only a 14 amino acid sequence, and it has great potential that I'm studying right now. So in this study, I have proved that this OGP is non-cytotoxic to the human periodontal ligament stem cells, or HPDLSs. Not only it's non-cytotoxic by proving a lot of these green colors instead of the red color here, but also it can increase the osteogenic differentiation potential to the cells, comparing with this control group by the evidence of the more red color in this line here. Not only that, we also found that the mechanism by regulating the TGF-beta pathway to regulate the bone regeneration. So by this mean, we know that this OGP has the potential to regenerate the bone tissue in our body or even in animals. Next is that we gonna incorporate this OGP into the hydrogel because we would like to make this OGP to become more stable and also to be slowly released to regenerate the surrounding tissue here. And this hydrogel not only to have that purpose, but it also can be injectable one. And that's why it can be less invasive and also more feasible to the clinical application. Thank you very much.
Okay, thank you, Stephen. Next for the Q&A session.
Not questions, Stephen. It's thank you for the presentations. It's not questions, but just a little bit suggestions. Pretty similar with the first presenters as well. Well, probably you could define the very technical or academic jargon, or probably more like how do we call it? Like medical term. So I don't know, like my principle is that even though you are a scientist or like academic, so once you are encountering a lot of people or audiences, they're coming from the very different backgrounds. So I was just hoping that you would be able to define a little bit further about what is actually osteogenic growth peptide in a very simple way before you go further to define or to explain other things. But I think overall the presentation is very engaging, and yeah, it depends on you. You want to answer my encounter or not. It's just very minor reservations from me. Thank you.
Yeah, thank you for the suggestion. So, well, actually, I made it up in this presentation. So, thank you.
Mas Stephen.
Yes.
Thank you for the presentation. So I just wonder when you finish this research, in which I mean, as you mentioned before, that this can be implemented in the human, even in the animals. But
Right.
In terms of accessibility of this final product, in which country or in which market do you think this will be? This can be your research can be accessed in terms of maybe the economic value.
Okay, thank you for the question. So, in terms of the accessibility itself, so I believe that this product, especially the OGP, is very accessible because right now I use the synthetic product from the brand. So yeah, it's very accessible to buy and it's very cheap because I use very low dose for this one. And in terms of the market right now, because yeah, our purpose to treat this periodontitis, the inflammation of the periodontal tissue, our oral health. So yeah, as I mentioned also, it can not only happen in animals, but also in humans. So our market can be broadened until the dentistry hospital and also in animal hospital to treat this disease. But still, because we still want to explore more, like to make it more visible of this product by using this injectable hydrogel. Thank you.
Thank you.
Okay, I would like to ask about for the next project you want to make this as a hydrogel. Why you need to make it as hydrogel?
Okay, thank you for the questions. It's interesting one. Why I would like to make it an hydrogel because first of all, the main product that I use is OGP and it's a peptide like protein, short one peptide. And we know like because peptide itself, if we just use it single compound, it may not be stable. It may be degraded during the treatment. So we would like to make it more stable, like slowly release as well. So that's why hydrogel would be preferred. And not only that, because it's hydrogel and it's 3D structure can be easily mimic the donor, the bone defect architecture. Thank you.
Yeah, thank you.
Okay, thank you, jurors, for the question. And thank you, Stephen, for the amazing presentation. Next, we will move on to the next participant, Priscilla Herma Yuriska. Are you ready?
Yes. Can you see my screen now?
Yeah, clear. Okay, the stage is yours, starting when you speak.
Okay, good morning, everyone. So I assume that all of you here are already familiar with PM 2.5, right? Yep, a teeny particle but dangerous. So this particle are so small and can enter and penetrate deeply into the human lung and also to the bloodstream, and it can cause the serious problem to human. And our study want to focus on understanding to how dangerous PM 2.5 in Thailand and what can be done about it. Especially, we estimate that the mortality that attributed to PM 2.5 exposure in short and long period. So why we need to do that? Because of the estimated number here matter when making decision for air pollution policy. So in 2019, the average level of PM 2.5 in Thailand was exceed the national standard as well as above the World Health Organization safest limit. And using the health impact analysis software based on the actual data of Thailand, we compare the health burden that relate with the national standard of PM 2.5 to the World Health Organization gold standard. We calculated that if Thailand can reduce the estimation of reduce the daily level of PM 2.5 to meet the WHO gold standard at short term of 50 microgram per meter cubic, and if the annual standard so can be reduced to meet the WHO world WHO long-term goals of 5 microgram per meter cubic, it means that we could prevent many mortality attributed to PM 2.5 exposure. So it means also the more people could be saved. So with this estimation, we provide the the finding to help the Thai policymaker to take action in order to control PM 2.5 and also improve the health of community. So everyone, when we take a break, it means that we inhale the air that can sustain our life, right? Not to harm, even not to reduce the time of our life. Thank you.
Okay, thank you. Next, for the Q&A session.
If I may start, hi Priscilla.
Hi.
Thank you for the presentations. Just give a little bit context. So I was living in Chiang Mai for years, and Chiang Mai has a very high level of PM 2.5, and I suffered every year. So I'm just wondering, I don't know what kind of PM 2.5 sources you are referring to, either coming from the factory or industrial sources, or maybe in the sources from Chiang Mai, which is slash and bird season. So that's my first observations. The second observations, and probably question as well, when you decide the sources of the PM 2.5, and then you say that you would like to communicate with the stakeholders or policymakers even in Thailand, I don't know how difficult it is, because in Chiang Mai itself, for example, we've been suffering for years. It's not just like 1 or 2 years, but every single year, always from January to let's say April or May until the PM 2.5 gone. So I'm just wondering how can you elaborate more or provide more explanations? How do you engage with the policy makers or stakeholders to deliver your finding, and how do you really put your research into actions at the end? Thank you.
Okay. Okay, thank you for the observation that you shared with us. And then yes, actually, Chiang Mai also one of the province in Thailand that also suffering with this PM 2.5 exposure. And then we also consider that in Thailand, characteristic of PM 2.5 and then how the increasing level of in many province is very different. Like in the upper part, like Chiang Rai in several area, it's maybe the cause is come from the burning forest or the burning of the crop residue, something like that. So like on the middle part, it's mean the concentration is come from the vehicle emission, something like that. So here's we consider that the level of PM 2.5 in the upper side, in the lower side, and in the middle part of Thailand is very different. And to ensure that our finding is can be applicable for the national standard. So here we just we just calculated and make the concentration of PM 2.5 to be the national. But actually, if you read on our paper, we also calculated for the province specific. But here, because of this is also part of the policy analysis process in Thailand, so we helped Thai policymaker to estimate the health burden. In the real project, actually, we have the several the cutoff or the standard that we use for short term and long term. And but here we just provide the current national standard that was announced by Thai government to be the national standard last year in 2023. And with this, we can help that the Thai policymaker can create a stronger action and also to make like a more targeted action related with the area that where the PM 2.5 is worse.
Okay, thank you, Priscilla.
Hello, Priscilla.
Yes.
Yeah, I just this is just my curiosity. Do you have any like comparative study between other countries, say our country, in your research? And as we may know that the level of this PM 2.5 also different between provinces. So my short question is, do you have any comparative study with other countries, maybe with Indonesia, and how do you find it? Thank you.
So actually we of course we compare as a background of us for to connect with this study, but here's mostly we compare to the world standard of PM 2.5, which is that provided by WHO, and also in Thailand itself they more like follow the process and also the regulation that provided by US EPA as a like they learn from them and have like a collaboration with that. But for our country, so far that sorry to say that hard to find the correct the current standard in our country. But I hope that in the future, when I learn this, I also can be like we also can adapt it and implement this process for Indonesia as well to get like the national standard and control the air pollution, which is also worse in our country, and also to protect our community.
Yeah, so maybe the challenge that I probably suggest you that we also need to do the same thing with Indonesia, then hopefully you can also disseminate what you have got to the government of Indonesia in future.
Hopefully in the future.
Okay, thanks for the presentation. I would like to ask, in your research, you do this by computational only or survey? Can you explain?
So this research use like a computational using the software that provided WHO that called RQ Plus. And in this software, we input the data based on the actual data of Thailand, like a mortality and then the concentration of PM 2.5 and the total mortality at the total population on this country. And also we put the cutoff value, or here we say as a counterfactual framework or the targeted national air pollution standard. Here we put like a 5 microgram for the long terms PM 2.5 and then the 50 microgram for the short terms. So yeah, and then we got the result that the proportion of attributed case that related with PM2.5 exposure, and also the mortality rate that related with PM2.5 exposure for the whole of Thailand and also for 77 provinces.
For a software use model or
Yeah, they have like a model, they have like a calculation
so it's
formula.
used from someone's model or someone's software or
Yeah,
model.
for WHO. Yeah.
Okay.
So we just put the data and also just run and get the result.
Okay, thank you.
Okay, the time is up for the Q&A session. Thank you, jurors, and also Priscilla for the amazing presentation. Next, we move on to the next presenter, Yusuf Arya Yudanto.
Hello, am I audible right now?
Okay, clear. You might start.
What about the screen?
Okay, clear.
Good morning, everyone. My name is Arya, and today I'll be delivering my posture. So it all began with the shift away from fossil-based materials, which is problematic to the environment. Bioplastics like polyolefin quickly gained popularity, and everyday add-ons like packaging and disposable products. But have you ever wondered what happens to these bioplastics once we throw them away? Yes, polyolefin is biodegradable, but as its popularity grows and more people use it, the waste it generates becomes a significant challenge. Simply relying on biodegradability is not enough. We need sustainable solution to manage this waste, and this is where my research steps in. Upcycling post-consumer polyolefin into foam via chemical recycling and supercritical CO2 foaming. Now, you may be familiar with mechanical recycling, where plastics are simply melted and reshaped. But this often leads to downgrading, where their recycled products loses quality and usefulness. In contrast, chemical recycling offers more advanced solution by breaking down the polyolefin chain into its monomer. This approach not only avoids downgrading, but transforms waste into something even more valuable like polyurethane film and blended foams materials with enhanced properties. To make this process more efficient, we use a microwave-assisted reactor as it provides rapid, uniform heating, significantly reduced reaction time and energy consumption. The product obtained for this process is called alcohol acetylyzed polyolefin, which serves as a valuable raw material known as biopolyol. This biopolyol is then utilized in the synthesis of polyurethane, and this approach enhances the circularity of polyolefin life cycle, promoting sustainable and eco-friendly utilization of the material. The resulting polyurethane is then blended with commercial PLA using an internal mixer to create multi-phase blend foams. The blended foam is created using a batch forming method that employs supercritical carbon dioxide as a physical blowing agent. This study successfully demonstrated the upcycling of post-consumer PLA into high-value bio-based polyurethane using microwave-assisted chemical recycling with the MPA as a terminating agent. FTIR analysis confirmed that the synthesis of urethane linkage evidenced by new peaks characteristics of these functional groups. When blended with the commercial PLA, the turkey profile displayed a consistent repeated pattern indicating a homogeneous blend for foam production. The resulting foam exhibits multifunctional properties, including a lightweight structure, adaptable compressive modulus, and open film morphology. This research highlights the potential for sustainability, high-performance materials through innovative recycling techniques, bridging science, industry, and policy to support biocircular green economy. Thank you for your attention. I'd be happy to answer any questions.
Okay, thank you. Juror might ask questions.
Okay, thanks for the presentation. I would like to ask a simple question. What is the difference between upcycling and also recycling?
All right. Thank you for the question. So recycling itself, it comes from the term cycling. So it can be downgraded or it can be the same level as the properties itself before recycling, and it's upcycling, which means the properties of the recycled products is enhanced from the raw materials. That's called the upcycling.
What properties you mean, like because in recycle, like we make something to be something else that can be used more also.
Okay, so like in this research, I use the post-consumer of polylactide. I break down the polymer chains of the polylactide into its monomer, and then I use that monomer as the raw main materials as a polyol to synthesize polyurethane. So then, like we can see from here, the properties that is enhanced is the mechanical properties and also the lightweight. So the goals here is to make the foam. So foam itself should be like it has the lower density because we want it to be like lightweight as it can. So when we compare to the like the general foam, maybe like polyurethane is actually made to be applicable for the foam, but this foam is actually we use the waste as the raw main materials. So we do upcycling. So we convert the waste into more valuable product. That's why we call it as an upcycling. We add value to that raw material. We add value to that waste. That's my answer.
Okay, thank you.
Thank you, Mas Arya. I have a simple question. I assume that there is also similar research available for the time being with your research. My question is, what makes your research different from the existing research, and what is what makes it different, and what makes it special compared to the existing research? Thank you.
All right, so thank you for the question. So basically, this research is part of the research center in my master program at Thammasat University. So in my case here, we use the DMPA as a terminating agent. So DMPA itself, it has a hydroxyl group and carboxylic acid in the backbone of their structure. So PLA itself is a hydrophobic. So we want PLA to be hydrophilic. I mean, like we want to increase the hydrophilicity of the PLA itself. So that's why we use the DMPA as a terminating agent, to be expected that we can use this as a for the biomedical application itself later on, such as for encapsulation or drug release, something like that. But in my case, in my research, I use this to make the foam, and then it has the potential application as a adsorbents for various organic solvents. And also, like from the graph, we can see that it has the enhanced for the mechanical properties itself. That's my answer.
Okay, thank you.
I think I'll give it back to Naila. I don't have any comments, so just appreciate your presentations. And I think I had a question as well, but it's already inquired by Ibu Daniela. So yeah, Naila.
Okay, thank you, jurors, and thank you, Yusuf, for the presentation. Next, we move on to the 11th presenter, Ahmad Effendi. Are you ready?
Hello. Hello.
Okay. Okay, audible. Okay, you can
Okay.
start.
Thank you so much. Good morning, everyone. Well, have you ever heard that there are more than 30,000 people hospitalized and 2,700 people died in the US due to the infections of multi-drug resistant of Pseudomonas aeruginosa back in 2019? Well, the infections of Pseudomonas aeruginosa have a wide range of severity, from acute to life-threatening, and it can be linked to practically any anatomical regions of the human body, including the skin. Another problem is that the Pseudomonas aeruginosa can produce a biofilm surrounding its colonization site. These film barriers obstruct the accessibility of antimicrobial agents to the bacteria. Considering the expected difficulties in developing novel antibiotics with acceptable side effect, an alternate way is needed to conquer this problem. Let's take a look at the left-hand side of the picture. You can see the bacteriophage or phage. Well, this creature is basically a kind of virus. I know it sounds scary, right? But you know what? This particular virus is so special because it can only kill specific bacteria without disturbing any other microbiota. That is why this creature has gained more interest nowadays. However, phages are very sensitive to the environment, making it way harder to use, especially for the skin-infected bacteria. Some research said that the topical application could not stabilize phages for a long time. Hence, it could decrease the effectiveness of the phage therapy. Moreover, the phages were not effectively delivered into the targeted area, especially beneath the skin. So, in our project, we have successfully designed detachable dissolvable microneedles made from hyaluronic acid combined with the gelatin that can incorporate and stabilize phage in for at least 28 days, stable at room temperature, or longer at lower temperatures. More importantly, the microneedles could also help deliver phages to the targeted site in the skin in less than 5 minutes of applications, with a painless, quick diffusion in the skin, which is way more effective than topical phage therapy. So, this project suggests that this microneedle could be used as a bacteriophage delivery into the skin. Thank you so much for your kind attention.
Okay. Next, for the juror, might ask questions.
Hi, Ahmad.
Hi.
Thank you for the presentation. It's very interesting. I just would like to know the level of your unit analysis. Is it in Thailand? And then how big is the unit of analysis? I'm just wondering because I think the way you present also deliver the ideas is like it's your findings actually applicable and also manageable to be spread out as wide as possible, right? I'm just wondering about the level of unit analysis, and that's it. Yeah.
So in my case, it's actually only in a lab in the lab, but this project is basically a branch of our a big project in our lab. So our lab has developed dissolving microneedle that can incorporate any kind of drugs, and we already have this kind of product. We already distribute actually in the market. So actually, you can find it in a marketplace such as like Shopee or Lazada, everywhere in Thailand.
Oh, okay, cool. Maybe you should present that product as well so you can also market your products here. But yeah, good luck. Thanks.
Thanks.
Okay, Mas Ahmad. Yeah, my simple questions actually, since your product has been marketed, so have I assume that you have measured the impact of the product. I wonder if you could explain more about the impact of your product.
Thank you so much for the question. So before answering that question, actually our lab is still working also in the developing of dissolving microneedle. So we have collaborator is actually kind of a startup company, and we made the product like the main product is dissolving microneedle. So this kind of microneedle can can incorporate any kind of ingredients. Like in cosmetics, you have like a retinol, and then in the drug, we already proved that we can stabilize and incorporate vaccines, but it's still in a clinical test. And for the impact, I think, yeah, because this product is still in the development, so I don't really know. But in the future, I hope this product can like bring a lot of functions, especially in a way to deliver something that are very beneficial to us. Yeah, thank you.
Okay, thanks.
Thank you.
I would like to ask about actually, is it like the band, like maybe Hansaplast or something different?
Yeah, it's, if you know, like the patch, acne patch, right? So it's kind of types of acne patch, but it has a very little needle inside. Yeah.
So it will cure like the swollen thing, or what kind of disease maybe?
So so far we developed the product in the cosmetical product. So it's for the disease we still working on it.
So it's like for acne.
Yeah.
Oh, I see. Okay, thank you.
Thank you so much.
Okay, thank you so much, Jur, for the Q&A session, and thank you, Ahmad Effendi, for the amazing presentation. Next, we move on to the next presenter, Iva Hafifah. Are you ready?
Yes, moderator, do you hear my voice?
Okay, clear. Cool.
Please allow me to share screen.
Oh yeah.
Do you see my screen?
Yeah.
Thank you so much. Good afternoon, ladies and gentlemen. Palliative care is essential for improving the quality of life patients and families facing life-threatening illnesses. In cases where the patient is in the end-of-life stage, it also helps facilitate a good death. According to WHO 2020, 56.8 million individuals require palliative care, but only 14% of patients worldwide receive palliative care. In Indonesia, there has been much research related to palliative care, primarily focused on cancer patients. However, what about the intensive care unit, which treats critically ill patients with high mortality case? Has palliative care been integrated into patient care in the ICU? The answer is that it has not been maximized. Based on the previous research, researchers conducted the quality of palliative care service in Indonesia in ICU setting. The result of the research, the palliative care in Indonesia, especially in ICU setting, at a poor level. The primary reason is the lack of understanding among ICU nurses about palliative care. And the other question is it the same as palliative care for cancer patients with the patients in ICU? This gap motivated the researchers to conduct a study of the concept analysis of palliative care in ICU in Indonesia. This study aims to identify the antecedents, attribute, and consequence of palliative care in Indonesia with the Walker's and Evans methods. Researchers found the antecedents include patients, family, healthcare professional aspect, and the attributes include wholehearted care for patients and families, culture and spiritual approach, interdisciplinary team, ethical issue, and decision-making process. And about the consequence of this study, not only for patients, but also family members, healthcare professionals, family members, and healthcare system in ICU in Indonesia. This study can provide significant benefits, not only theoretically, but also practically. This study can enhance palliative care service in Indonesia as healthcare professionals, particularly nurses, develop a better understanding about the meaning of palliative care in the ICU and factors influencing palliative care in ICU. This understanding will enable nurses to deliver care more effectively, thereby improving the quality of life for patients and family members, and ensuring the patient can achieve a good death. Additionally, the findings of this study serve as a foundation for the development of qualitative and quantitative research related to palliative care in ICU in Indonesia. Once again, palliative care in the intensive care unit is very important. Palliative care have a beneficials not only for patients, family members, healthcare providers, and healthcare system in ICU in Indonesia. Thank you so much for your kind attentions. Good afternoon. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Next for the Q&A session.
Hello, Eva.
Hello.
Thank you for reminding us that there is a good life and good death. I just have a questions regarding the explanations about the misunderstanding, like why nurses, why healthcare people who really have to have some kind of understanding about this, because it will affect not only to patients, especially the ones who have a critically ill condition, but also the patient's family as well. I'm just wondering, where do you see the very roots, the very foundations of the issue in Indonesia, for example, why they don't have, especially the nurses, they don't have understanding about this kind of understanding? Is it because of the curriculum of their nursing studies, for example, or it's just some kind of, it's not really integrated to our healthcare system, or how do you find that?
Thank you so much for your questions. It's very nice questions. Yes, the palliative care, the first WHO explained about palliative care. Palliative care in 1990, but the definition of palliative care, especially only for terminal illnesses or end-of-life patients, but also the majority of nurses understanding palliative care only for terminal illnesses. But WHO explained the new definition about palliative care in 2002. The palliative care not only for terminal illnesses, but patients have a diagnosis with the life-threatening disease. Not only patients, but also family members require palliative care. So the majority of nurses not have the good understanding about the new definition from WHO, especially in ICU nurses, because in ICU nurses have the mindset for focus to treat to give the intervention only for critical illness to life saving. But also for palliative care, the focus not life saving, but give the comfort for patients, like free from sufferings from patients until the patient death, so patients can achieve a good death, and family members have support from in the bereavement phase or grieving phase. This is the reason. So a lot of ICU nurses not have the good understanding about the palliative care in ICU. So the researchers have conducted this study to explain what is the really meaning about palliative care in the ICU setting. It is different with the other settings. Thank you so much.
Okay, cool. Thank you.
Okay, Mbak Eva, thank you for your presentation. I wonder that from your literature say that the palliative care is still poor in Indonesia, if I'm not mistaken. So how convinced you are by having this research it will improve the palliative care in Indonesia, and what method will you implement to make the palliative care in Indonesia better from your research? Thank you.
Yes, thank you so much, Ibu Daniela, for your questions. It's very interesting questions. My study focus on the palliative care in the ICU setting. But also in Indonesia, a lot of study about palliative care. But the focus of the previous research is on the cancer patients or terminal illnesses like tuberculosis, stroke, and etc. My study focus in the ICU setting. It is a lack of study about palliative care in ICU setting. This study is part of my dissertations. In the preliminary research, researcher conduct the concept analysis first to know the meaning, the exactly meaning of palliative care in ICU, because it is different with other settings. So after this research, we will continue to explore what is the factors influencing the palliative care in ICU in Indonesia. And then the last project of the dissertations is to develop the intervention to increase the palliative care service in ICU in Indonesia. So all of the research is very important to enhance the palliative care in ICU in Indonesia. About the researcher statement previously about the poor quality of palliative care in Indonesia, it is the study focus on the palliative care in ICU. But palliative care in Indonesia is a good level, but focus on the cancer patients. In the ICU, it's still a poor level because researcher conduct several research since a master degree until now, focus on the palliative care in the ICU setting. Thank you so much, Ibu Daniela.
Okay,
Do I still have time?
the time is up for Q&A session. I'm sorry.
Okay.
Okay. Okay, thank you so much
Thank you so much.
and also Ifah for the presentation. Next, we move on to the next presenter, Yoga Saputra.
Yes.
Okay, are you ready?
Can you see my screen?
Yeah, it's clear.
Yes, and my voice is audible, right?
Yeah, audible.
Okay.
Start.
Good morning, everyone. Today I'm gonna present my research in synthesis of tungsten oxide supported on iron oxide as a magnetically retrievable catalyst for selective oxidation of styrene. So the background is this research is benzaldehyde. So benzaldehyde has been known for many years, and it has been used in many industry like perfume, pharmacy, sweetener, and many industry. But unfortunately, the traditional method of synthesis benzaldehyde involving harsh condition which required high temperature and high pressure. In this research, I want to give an alternative way to synthesize benzaldehyde from styrene using magnetic retrievable catalyst. The goal is I want to synthesize the streamless purification process, the catalyst that easy to separate, and then selective catalyst, and then the catalyst that can be used for several times. And of course, the wet condition reaction, which is low temperature condition. The synthesis method of the catalyst, I use the co-precipitation method, and I also synthesize the iron oxide. And after that, I calcinate the materials, and I use the material for the reaction. And for the reaction result, I use gas chromatography to analyze how much benzaldehyde I get from 1 millimole of styrene. And I also study about the optimum reaction condition, which temperature I study from 60 to
to 90 degree. I also study the optimum time. I study from 1 hour to 24 hour. I also study about the oxidant, 4 types of oxidant, and the ratio of each metals, the recyclability, and the optimum amount of catalyst, optimum for producing benzaldehyde. And I also characterize the catalyst. So I study the morphology of the catalyst, the crystallinity of the catalyst, the surface area, the oxidation size, magnetic property, of course, because it's a magnetic catalyst, and I also study about the size, shape, and morphology. From the characterization and the optimum condition that I get, I can conclude that tungsten oxide supported on iron oxide is a promising candidate as a catalyst for benzaldehyde production. And the catalyst demonstrates good selectivity up to 80% and compression up to 60%. The catalyst is recyclable up to 5 times, and effective is the catalyst can be used in the low temperature, 60 degree. And the catalyst, the main goals of the catalyst is that the catalyst is easy to separate from the reaction mixture. Thank you so much for your kind attention. This is all of my presentation.
Okay, thank you, Yoga, for the presentation. Next, the juror might ask questions.
So thanks for the presentation. I'm just wondering what's the problem that you face when you do this research? Problem.
The problem is I quite challenging to study about the optimum time and the temperature. So I study about the optimum time and then 24 hour. But when I study the temperature at 24 hours, 60 degree given the good result. But 70 degree is given the lower result compared to 60 degree. But when I study this temperature, 70 degree is giving the good result. So the first challenge that I face in this research is that I have to prioritize which optimization that I need to do first. So I must admit that I'm lacking on that part. So it's become how to say, is a challenge of the study at the beginning when I start to studying the optimum condition.
Okay, thanks. That's from me.
I think I have my notes already, so I'll give it to Ibu Daniela instead, Ibu.
Yes, thank you for your presentations. I just wonder the level of your confidence that your research will be implemented in a big scale. Maybe you can elaborate more about that. Thank you.
For the in the industrial scale, I am confident that the catalyst can be produced in the mass scale. But because this catalyst is studied in the chemistry, I mean the field of chemistry, I need to collaborate with the engineer chemistry to design how the reactor will be, and the other aspect that is need to be optimized in the industrial application, because is in the lab application is quite different in the industrial application. But I optimist that the catalyst can be produced in the mass scale, and use in the industrial application, because the goals is also in the goals is also one the catalyst to be used in the industrial application.
Okay, thank you.
Okay, is there any more questions from jurors, or can we wrap the Q&A session?
I think we can wrap up, Naila.
Okay, thank you so much. And thank you for the presentation, Yoga. Next, we move on to the next participant, Emi Erawati. Are you ready?
Okay. Good morning, everybody. My name is Emi Erawati. I am PhD student in the Department of Integrated Chemical Engineering, Faculty of Engineering, Maidol University. Today I will present. The title is Application of Iron Oxide, Silica Oxide, Zinc Oxide, Magnetic Composite as a recyclable heterogeneous nanocatalyst for biodiesel production from waste cooking oil, response surface methodology. Iron oxide, silica oxide, zinc oxide nanocomposite was synthesized using the co-precipitation method and then used as an efficient and reclaimable heterogeneous catalyst for biodiesel generation from waste cooking oil. The sulfur feature of nanocomposite were determined with X-ray diffraction, Fourier transfer infrared, mapping ADX, phase emission scanning electron microscope, transmission electron microscope, and vibrating sample magnetometer. The specific surface area, mean particles, and pore volume of iron oxide, silica oxide, zinc oxide were estimated to be 32.65 meters square per gram, 10.5 nanometer and 0.5 centimeter cubic per gram, respectively. The result of nanocatalyst characterization indicate that the distribution of silica and zinc element in the lattice of iron oxide nanoparticle has been done successfully. The response surface methodology and bulk banking design were applied to design the experiment, statistical modeling, and optimization. The R-square value for response surface methodology was found to be 0.99, which indicates that the model is remarkable, consistent with the experimental data. The maximum biodiesel yield for the synthesized nanocatalyst was achieved at 97 under optimum condition, including methanol to oil molar ratio, ultrasonic irradiation time, and nanocatalytic dosage were 90.9 to 30.1 minute and 2.6 the percentage of weight. The stability assessment illustrates that the catalyst can be efficiently reutilized for 7 cycles. Furthermore, it can be concluded that the iron oxide, silica, zinc oxide nanocatalyst can be applied as an efficient and promising heterogeneous catalyst in biodiesel generation from waste cooking oil. Thank you very much.
Okay, thank you, Emirawati, for the presentation. Next, the juror might ask questions.
Hello, Amy. Thank you for the presentations. I have a questions regarding the when you said about the waste cooking oil. So is it coming from the household waste cooking oil or industrial ones? It's like hotel industry or maybe restaurants, etc. Just wondering about that level analysis. And then the second one is, how do you see after you have done this research? Let's say that I'm just trying to foresee from the some kind of interdisciplinary perspectives. After you've done this research, and then what is your expectation to apply some kind of finding from the research, if you get the points?
Okay. Thank you very much, Ms. Hannah. The waste cooking oil was collected from canteen because this is a paper written by Basir Maleki and Hossein Ismaili. This is collected from canteen in Iran. The research is very important because waste cooking oil is waste. We can treatment the waste become fresh biodiesel from waste. So it is can reduce waste through to sea or lake and river, and also if we treatment waste cooking oil can reduce the emission to environment, because the pollution with waste from oil is lower than from diesel fuel.
Just quick clarification. So this is the presentation is coming from others researchers, right?
Yes.
Okay, got it. Thank you.
Yes.
Thank you, Mbak Amy. Just simple questions from me. Maybe I just want to have your opinion in Indonesia setting. As you know that now we would like to produce the biodiesel from the palm oil that now happening, and I think this is one of the priority programs from the current government. I want to have your opinion. How can we connect your research with the Indonesian setting for the time being? Thank you.
In my opinion, my research can apply in Indonesian country because in Indonesian country, because the production of biodiesel developed from first generation. In the first generation production of biodiesel using raw material for food, so become second generation. In the second generation, production of biodiesel come from non-edible oil based on the president obligation side. In this year, president will increase the ketahanan pangan. So if we use first generation, in the first generation, the raw material of biodiesel come from edible oil. Edible oil is suitable for food. So if we using sorry, it is enough.
I think you can go ahead. I mean,
Okay. It is finished for me.
you still have time, I guess.
Okay.
There's still time for the Q&A sessions. Do you still have any questions, juror? Okay, I think that's all for the Q&A sessions. Thank you, Amy, for the presentation. Next,
Thank you
we
very
will
much.
move on to the next presenter, Bion Nugraha Jayawiguna. Okay, are you ready?
Okay, wait a minute. Okay, are my sound and the camera working properly?
Yeah.
Okay. Okay, good. Yeah, okay, good. Please let me know if you can start.
You can start now.
Oh, okay, yeah. Okay, so Assalamualaikum. In this 3MT event held by Permitha Days, actually I'm scheduled to present towards the very end, so I hope the judges and the other participant still enthusiastic to listen, especially in my work. So I'm here talking about gravity in theoretical way, which might sound complex, but we can break it down in a simple way, hopefully. So we can start with instead of the attraction forces like Newtonian gravity did, Albert Einstein in 1915 explained that gravity is a bending of space-time, and we can illustrate by imagining the big trampoline with a heavy object placed in the center. So the trampoline stretches, creating a deep curvature. Then we can take a smaller object and rolling around, and we can see that the bubbles start to spiral, and that is how gravity works in Einstein gravity, and that is how our solar system works. And mathematically, it expresses like this. In the left-hand side, we have the pure mathematics aspect, and the right-hand side can have a physics aspect. And the solution of the Einstein equation is the relation is mass and the radius relation. This is the solution of the field equation. And you can see that the photons in the red line here, red area here, is the photon sphere where the gravity, the gravitational wave or the ripples of space-time could happen. But no one exceeds this kind of region with nowadays. So in my research, by adding a little tweak with in the equation, in the Einstein equation, we wondered if we can push beyond the Einstein theory to exceed the photon sphere region. So it is aligned to our motivation that we try to obtain the suitable model so that the mass and radius can exceed the photon sphere region. That is our motivation, and we start with the star solution accessing the space-time in Einstein theory, which is denoted by the beta. The beta is the modified parameter, and this contribution is to increase the radius of the star. And then from that moment, we can see finally that the mass of the star can exceed the mass and radius, the photon sphere region, because with this fixed R, the M is 3.1 times mass of the sun, which is inside the photosphere. So the gravitational wave exists, and depicted is in the right figure, this one. And our modified model is in the red one and has a smaller value due to the smaller in the effective potential, so that the wave that come inside of the star, they can oscillate back and forth inside the well of the potential quickly. And these are my reference, and this research is supported by C2F scholarship. Thank you very much.
Okay, thank you very much, Bion, for the presentation. Next, the juror might ask questions.
Okay, I would like to ask the question, and thanks for the presentation. Yeah. What do you mean by gravitational wave here? And why it appears or the gravitational wave can be exhibited from the photosphere.
Okay, yeah. So the gravitational wave is like an actual wave, yeah, happening in the Newtonian gravity. So if we can imagine that trampoline, if we have a heavier object and creating a heavy object placed in the center in the trampoline, we can see that the trampoline stretches, creating a very deep curvature. And that is the Einstein gravity point of view. But the gravitational wave is that when the star or the heavy object start to move, they will get accelerated, and the wave have a ripples, and then the ripples will go outward. So that's why it's called gravity. That's why it's called wave in gravitational theory, because this gravitational theory, the gravitational wave is the first initiated by the Einstein theory. And for the gravitational wave in the photosphere, it's quite makes sense because the photosphere region has the bigger mass than the usual star. Because if we can see the several black line, this is for the smaller mass. But if we can go high enough mass, we can go to the photosphere region, and the curvature around the trampoline will get bigger, so they will go deeper. So that's why the gravitational wave is only appear in the photosphere region, because they should have a bigger mass. I think that's the answer, sir.
Okay, thank you. That's from me.
Yeah. Okay, thank you very much.
Beyond, thank you for your presentation. So my questions may be around the impact of your research. So maybe if you can elaborate a little bit about what it means in real life.
In real life, okay.
Thank you.
Okay, yeah. Thank you very much for the very enthusiastic question about the motivation in real life. Okay, but I think we can start with the black hole, that actually mysterious object in the universe. But in 2015, it appears experimentally, while the theory is quite established in 1915. So it's more than 20 years to prove the existence of the black hole and the stars like this profile. And the black hole, the black hole object in this Einstein theory, there is no interior inside of the black hole, and this impact, this existence of the gravitational wave, the impact is that we can try to find the interior of the black hole, because the wave, the gravitational wave, comes from wave that comes inside of the star. So the response that go outward is the information about the mass and radius. If we get the mass and radius exactly the same as the black hole object in the universe experimentally, then we can conclude that we find the composition of a black hole. I think that is the quite realistic answer.
Thank you for that.
Do we still have time, Naila?
It's still there's still time.
Okay, hi Bjorn. Thank you for the presentation. It's very interesting. I understand fully that you have different background with some audiences, including me as well. But I just want to clarify a little bit further, correspond to what Ibu Daniela was asking earlier. So, who is actually your main target of the research. I think it's okay not to be applicable for society as a whole, but I think it's really useful to state first thing first, like probably my research contributes to the, for example, like NASA scientist. That's okay, but as long as you state it, so people will not have high expectations for that. Thank you.
Okay. Yeah. Okay. Thank you for the question. Okay. So my point is, we have 2 points. The general motivation. The first one, we want to try because these several backlights is the star profile obtained from the experiment, and no one exceeds the photosphere region, right? So my first motivation is that we want to find a suitable model that can exceed the photosphere model, so the gravitational wave will exist. That is the first one. The second one, if we have a model that already in this area, we can improve the density model so that they can go bigger, so that they can exceed the black holes region. So that is the general motivation. So in the second one, in short, we want to try find the interior of the black hole, because until now, no one knows the interior of the black hole. And so many proposals improve the model, but some of them are not successful. I think that's all.
Okay, cool, and good luck for the work. I give it to Naila.
Okay, thank you so much.
My pleasure. Thank you very much.
Okay, thank you very much, the juror and also Bjorn for the presentation. Next, we move on to the last presenter, Ismah Lutfia.
Hello, everyone. Are everything is still here now?
Okay, yeah.
We're all here.
Okay, are you ready? You can start screen sharing.
I'm ready, but why? Can you see my slide now? Can you hearing my voice now?
Yeah, it's clear.
Okay. Okay, I hope everything is still wake up and enthusiastic to see my presentation. Okay. I will start now. Good afternoon, everyone. Okay. Oh, actually, I want to talk about my research. Imagine a world where electric vehicles charge quickly, run longer, and environmentally friendly in the future. This is not a distant dream, but a tangible reality made possible by groundbreaking advancement in the carbon quantum technology, electric motor development. Today, I will introduce you to the carbon quantum revolution, CQR EV, transformation advance in electric motor development for supercapacity integration. Okay, one of the key points, Indonesia is one of the countries with abundant coconut carbon waste. According to the FEO data from 2021, Indonesia ranked as the one of the largest coconut producer in the world, with an annual production of approximately 18.3 million tons. The waste from the production, such as a coconut fiber and shell, is often not utilized effectively, posing significant environmental challenges. That's why in here, I want to bring it in the carbon quantum revolution, CQR EV. This can utilize the coconut waste to the materials with the high performance suitable of the supercapacity. In my research, I want to focus on the optimizing the surface of the structured quantum carbon materials to maximize the energy storage capacity and ion diffusion efficiency. This is the key point. If we want to depend of the energy storage, we needed to get more higher the capacity, more get higher the efficiency. So my experiment target, I want to make electric motor integrated with optimized supercapacity can achieve higher output power, greater operational range, and faster charging compared to the traditional EV. So in here, the carbon quantum revolution, CQR EV, demonstrates how the technological innovation can address two critical issues. For the one is it can get more enhance the efficiency of the electric vehicles and mitigating environmental waste by harvesting carbon from the coconut waste can create greener and more sustainable in the future. Okay, thank you very much.
Okay, thank you very much, Isma, for the presentation. Next, the juror might ask question.
Thank you, Mbak Isma,
Yeah.
for the presentation and for your enthusiastic presentations. So I wonder if you are from the existing research, what do you find about the existing research that may be similar to what you are doing right now? What area that you would like to improve based on the existing research that you find in your literature review?
Okay, actually in my literature review, it's we talking about the quantum. So I know that everyone have different background, but I want to tell a little bit about the quantum. Quantum, it's like the theoretical, but it can get more higher capacity with the same amount. So if in the traditional EV, usually we just can save 10 farad, but if we using the quantum technology, we can create more. It's already get 100 farad or something like that. And from the literature review now, a lot of researchers still doing the methodological, but don't know how to make commercialization. So in my research, I focus in the commercialization step because my background, the last time my bachelor and my master, I'm already doing the researcher, and my PhD now, my PhD now, I want to learn how to making commercialization, because like we know that like nickel in the Indonesia, we have a lot of forge, but we still don't know how to manage it with a good way with the commercialization like that. Yeah. Yes. Miss Daniela, thank you for your
Thank you. Thank you.
Hi, Ishma. A very good energy. Thank you. Thank you for the presentations, by the way. I just would like to probably asking clarifications. For example, if you mentioned, so you were comparing between your research and then the energy that we've been using for the so-called conventional electric vehicles. I don't know your observation so far, but I think in Indonesia itself is kind of new for having EV, right?
Yes.
So I'm just wondering how, and then your focus is more like the market system, marketizing the product. So I'm just wondering how do you see your research can compete with the EV in Indonesia, and especially when you try to utilize is it coconut shell waste, right?
Yes.
The shell, right? So if I'm not mistaken, Indonesia also has been exporting coconut shells and also kernel shells as well to Japan at least, and also South Korea. So I'm not sure how do you see the competition between that instead of producing our own energy or just instant income from exporting the coconut shell to other countries. Yeah, just would like to know your perspectives on that.
Actually, I want to explain a little bit. Actually, I want to talk anymore, but we don't have a lot of time. But a lot of researcher in energy storage, it's still try to find a good materials because we cannot change the lithium, right? We have limitation in the lithium. We needed to find the new source material and don't have a limitation. That's why I want to focus in the coconut shell and I want to focus in the nickel. That's why the quantum technology, it's not depend from the materials, but it's depend from we can make from the material waste to have a new technology or structure, so the performance it's more higher. I'm sorry. So your key point, it's can I get the key point of your question?
Sure. I just would like to know your perspectives about how you marketize your product, since it's also a new thing for EV itself. Indonesia is a new thing, let alone using this so-called new energy.
Actually, a lot of researcher energy storage is doing just focus in the battery of lithium because the performance is so high. And I know that we must know that we have a lot of source actually in Indonesia. I want to bring it because if I go to conference, meet with everyone from another country, everyone will asking about can I use your source from Indonesia and how you manage it, how the regulation, something like that. But everyone don't know about how to can use in Indonesia because regulation in Indonesia have a lot of problems, something like that. And no one, everyone from another country, it's still difficult to talk with Indonesia people because the regulation it still don't have any detail. Like we don't have the regulation how to another country can use it and what the what we have thought it's so impactful in the future. And my key point, I want to making the materials with the high source in Indonesia have impactful and how I can marketize. That's why I come to SUT because SUT have a good about the business model and how I can marketize large up my idea or something like that. Yeah, but now I already doing that. That's why, yeah.
Okay, good luck, Susu, for your work.
Yes, Permitha.
Okay. I have some questions. First, what kind of quantum material that you use here? Because you extract from the coconut, right?
Right. What kind? You mean I must talk the detail method?
Just the quantum material. Because we know that lots of material.
Yes. Oh, sorry. Again, please, I don't get the point.
So in your battery, you will use the quantum material. What kind of quantum material? Because we know that so many material that can be used.
Yes. What kind quantum material is carbon? Carbon-based coconut. So the quantum it's make a new way from the structure. So like if we study about nanoparticle, we just can keep it the energy storage in the nano value, but in the quantum from nanoparticle our material we can making new way, and so the amount of storage is more higher too. The key point of technology quantum is like that. So we can talking like we have analog and digital, and the quantum it can save more higher than analog and digital. What kind is it?
So you
get my point, or
so you will make a battery from carbon?
Hi, from the coconut cell, I make new structure, modifying the porous. I add more the quantum properties inside my carbon. After this, I can use in the battery. Actually, in the battery we have cathode and anode. Usually in the battery, anode is using graphene or graphite, right? And my focus using the supercapacitor. That's why I use the biomass or activated carbon base.
So the quantum is on the capacitor.
On the supercapacitor for the cathode, and on the battery to be anode. So the quantum is from the carbon, we can be activated carbon, we can be graphene, we can be graphite. For the activated carbon, we use in the supercapacitor device. In the graphene and graphite, we use for the battery device. Actually, energy storage have two. For the one is supercapacitor and two is the battery. We can use all. That's why carbon is so promising in the future because we can get market not just from the supercapacitor but in the battery. The battery, the kind it's anode. The cathode we can use lithium, but the anode is still important thing. We needed new source on that. That's why I focus in the carbon, because can both, not just one device.
Okay, thank you.
Okay.
Okay, thank you so much, Juror, for the questions. And thank you, Isma, for the amazing presentation. And that wraps up our competition 3 minutes thesis today. Before we leave the Zoom, I would like to thank you all. Thank you for our amazing jurors, Bu Daniela, Kak Fitra, and Kak Hana. And also, I would like to thank you for all the presenters and the audience that is still here with us. And before we leave the Zoom, I would like to take a documentation together. You can open your camera and prepare for the best post. Okay. Is there anyone that still want to open the camera? I think that's all. Okay, I will count 3, 2, 1. Okay. One more. 3, 2, 1. All right, cool. Okay, before we close our event today, I would like to remind you that the winner will be announced tomorrow in puncak acara Permitha Days at KBRI. And we will also have seminar wawasan kebangsaan, so don't forget to come tomorrow. And thank you everyone for joining. See you tomorrow. Bye-bye.
ครับขอบคุณครับเจอกันพรุ่งนี้นะครับ
ขอบคุณค่ะ
เจอกันอาทิตย์หน้า